Welcome, everyone. Thank you so much for coming together to celebrate the research work that the first-year research experience and summer undergraduate research fellows have been doing this summer. And Kevin, would you like to say a few words?
Yeah, we're happy. And I think, Lance, correct me if I'm wrong, this may be the inaugural kind of end of summer gathering, kind of a works in progress or works in completion kind of mini symposium for CURE. And so we're really excited to have all of our amazing students with us and their faculty mentors and other supporters and friends. And so thank you for joining us. We had our McNair Scholar Symposium this morning, so it's been a day of symposium. So again, it's been a privilege to get to know and work with the students over the summer. Lance has been leading our kind of check-in meetings throughout the summertime, and we all recognize it's been a challenging summer for many reasons. The pandemic in particular obviously has exacerbated some of the challenges that research often brings with it. I know access to labs, research sites, and other things have been very difficult. And so we just want to, again, acknowledge your adaptability and creativity and the wonderful support from all of the faculty mentors. And again, it's just been our privilege to help be a small piece of supporting your work as part of the Center for Undergraduate Research Engagement. So again, thank you for joining us. And Lance is going to be our pilot. I will be sharing the screen sharing privileges, but Lance is going to be our MC and pilot today. So I'm going to turn things back over to Lance.
Thank you. Okay, so let us, so I've dropped the schedule of presenters again in the chat, and let's kick off with Myles. Why don't you introduce yourself and give us the highlights of the work that you've been doing this summer?
Awesome. Well, let me go ahead and share my PowerPoint. Does everyone see that?
Yes.
Awesome. So my name's Myles Nelson, and my project is predicting the metabolic cost of level ground walking from gait speed and prosthesis stiffness. And my mentor is Professor Mike Hahn in the Department of Human Physiology. And so just a little introduction. So each year in the US, an estimated 150,000 individuals undergo lower limb amputations, and prostheses are intended to restore locomotion, but individuals with lower limb amputations still experience mobility limitations. And so research shows that elevated metabolic demands while using a prosthesis are common among users, and increased user specificity may diminish elevated metabolic demands, specifically the foot prosthesis stiffness. And so from a connection at the University of Wisconsin-Madison, this is a variable stiffness foot prosthesis. And so my project is to be able to predict the metabolic cost of an individual using this foot prosthesis based on their gait speed as well as the stiffness of the foot. And so most prostheses are a single stiffness, which doesn't really allow for the users to have great range of motion as well as speed. And so an example I like to say is if you're late for a bus and you need you're a little late, you might go from a walk to a brisk walk to maybe even a jog, depending on how late and if that bus is starting to take off. And with a usual single stiffness prosthesis, you might not be able to go all those three speeds without really increasing the energy you need to run that speed. And so with this foot, it would change the stiffness to allow you to walk at different speeds, maybe that jog or that brisk walk, while not elevating your metabolic demands. So that's kind of what my project is about. And this summer, well, this is the methodology. So I'm going to be using 15 healthy able-bodied individuals. It's a 5x4 system, 5 stiffnesses with 4 different velocities. And then we'll collect the VO2 and the heart rate. With COVID, we weren't able to do that this summer, and which was unfortunate, but the lab is now open and I'm getting started either this week or next week. So I'm super excited about that I can actually start the data collection process. But this summer, I was able to use pilot data and did a bunch of coding, both for analysis. So I was able to use that pilot data to kind of practice my analysis. That way as soon as I'm done with data collection, I can just run it into this code and it'll spit out all the figures I need, as well as all the information I need to create my predictor equation. And in addition, I created additional code that will connect the foot prosthesis that was on the earlier slide directly to my laptop, so I can control it from my own laptop instead of additional equipment in the lab. And this will just make it a more seamless process, as well as give data directly to my laptop instead of additional computers in the lab, which will just make it so much better for me and quicker to analyze everything. So that's what I did this summer. I'm really looking forward to getting started with actual data collection now that things are opened up, and I got approval from everything that I need, and I'm starting to get subjects lined up, and so really excited to get that going. So thank you, and if you have any questions, I'm happy to answer them.
Thank you, Myles. Any questions for Myles? Well, if you do have questions, you're more than welcome to put them in the chat. So Nicole has not arrived yet. So Jude, are you queued up to go? And then once Nicole arrives, she can speak after you. Does that work?
Yeah, definitely.
Fantastic. Okay.
Okay, Jude, I just shared the privileges, so you should be able to share your screen.
Okay, great. Thank you. Okay. Can everyone see that? Okay, great. So hi, my name's Jude, and my thesis is titled Racial Influence on Transgender Hate Crime Verdicts. So the primary research question that I'm looking into is looking at how race influences the convictions and hate crime charges that are applied to the murders of trans femme people, depending on both the victims and the perpetrators' races. So to break that down a little bit, by trans femme people, I'm breaking people into two large categories of transgender women and gender nonconforming people, which here I'm defining as people who were considered male at birth, but who were presenting effeminately when they were killed, although they didn't necessarily identify as women. And I'm breaking races into 5 large categories. I'm using both state and federal cases, and I'm looking at every murder case brought in which the victim was a trans femme person since 2009, which is when the Matthew Shepard and James Byrd Jr. Hate Crimes Prevention Act was passed. And I'm using a variety of political identity theories to examine my data. And for resources, I'm primarily using news reports and court briefs. So some limitations that I've encountered so far are that when trans people are murdered, it's often underreported or misreported. And also, the government hasn't actually conducted any statistical research on the oppression that trans people face, both in government and legally. So often trans femme people are discredited because their evidence is anecdotal. And also a lot of researchers have neglected intersectionality, which means, for example, they might focus only on gender identity or on race without looking at both, which means that people like trans people of color who experience multiple marginalized identities like that are left out of the equation. And then also that race has to be determined by eye, right? So I'm looking at images of perpetrators and victims to categorize them in race, but I obviously have my own preconceptions and biases about what race means and how it's defined. And likewise, so do the jurors who are looking at perpetrators and victims to determine their races as well. So that's inevitably subjective. So I'd love for you to note the identities of, see if I can move that, of the people in these statistics. So 42% of trans people in America have been physically attacked in their lifetime, almost half. And then more than 8 out of 10 trans women who have had to interact with police have experienced verbal, physical, and or sexual mistreatment. And of the trans women or trans femme people who are murdered every year in America, 91% of them are Black. So we can see that when these marginalized identities are stacked on top of each other, these people become even more targeted by oppression. And importantly, 17% of jurors who took a national survey admitted that they couldn't actually be impartial when ruling on case subjects involving LGBTQ+ people. So I just wanted to show you my data as well. This is what the table looks like of how I'm collecting information. On the left is demographic data that I already talked about. And then On the right, I'm also collecting things like method of violence that was used, what defense was used, if it was considered a hate crime legally, things like that. And the biggest thing that I found so far is regarding pleas. So when someone is accused of murder, they can, broadly speaking, plead guilty or not guilty. And if they plead not guilty, they go to trial. Most people, so on the left, that's the national data, and on the right, it's my data specifically from my research. So most people plead guilty because they don't want to risk going to trial, because most people are found guilty in trial anyway. And if you're found guilty in trial, you generally get a harsher conviction than if you had just pleaded guilty originally. So 70% of people plead guilty nationally, but when the murder subject is a trans femme person, less than 40% plead guilty. And I know you can't see this statistic right here, but it's 58% that plead not guilty. So what that means is that perpetrators are nearly 4 times more likely to risk going to trial when they murder a trans femme person, and they're over 3 times more likely to be acquitted of murdering a trans person. So what I'm arguing in my thesis is that it's because trans people are so dehumanized in our society that jurors have less sympathy when the victim is trans, and therefore they're more likely to acquit a perpetrator of that crime. But I also want to problematize what we consider justice to be for these trans femme people in my thesis, because while I'm arguing that trans femme people, especially of color, are seen as deviant, it's also important to acknowledge and look into the fact that men of color are seen as hyper-violent. And so when they do happen to be the perpetrator, juries also aren't sympathetic to them as well. So it's a tension of whose body is being policed more, almost. And then I just wanted to end by acknowledging these are the trans femme people of color that I've encountered so far who are not in the data table because the person who killed them didn't even have charges brought against them. So thank you.
Thank you so much. Really, really impressive research. Does anybody have a question for Jude?
Yeah, I have a question for Jude. I was wondering if you, in your research, noticed any geographical patterns when looking at these cases, if there are concentrations in some areas of the United States where it's higher, or whether it's, in more urban populations versus rural, if you've seen any patterns there.
Yeah, I've noticed I still have a lot of different relationships between the data that I'm going to look at, especially specifically racially. But I have noticed, I guess this would be more anecdotal because I haven't specifically gone through it, but it's definitely the cases that happen in rural areas are much less likely to have charges brought against the people. So they're more likely to be on that list I showed at the end. And I've also noticed, which I think just goes with population, that kind of the bigger the city is, right, the more people who end up murdered, which is true across the board. But I have noticed that in the South, there tends to be a lot more specific cases. And interestingly, it's specific cities too. Like there's a lot in Atlanta specifically, for example, or Tampa, where Black trans women have been murdered time and time again within a 5-year stretch.
Wow, thank you. Anyone else? Okay, I see that Nicole Mullen is here, so let's queue you up to go next. Thank you so much, Jude.
Wonderful. Okay, Lance, are you going to enable, so then I'll share my, okay.
You should, yeah.
Okay, here we go. Okay, can you all see that well?
We can.
You see that beautiful, all of those? Okay, so that right there is a bit of a compilation of some of the Zoom calls that I made with people from around the world about philosophy with children. So that was what my grant was about. I applied for the grant to learn more about doing philosophy with children and what kind of educational value this had. My original research question was just, what is the value of doing philosophy with children? But as COVID-19 swept through and conferences got canceled and things like that, the question actually morphed a little bit more into what is the value of doing philosophy with children, especially during moments of crisis. So the moments that I focused on were COVID-19 and the movement for Black lives. And then a caveat of that as well was, how does doing philosophy with children change with technology? Because of COVID-19, I was on my computer a lot, and so were others. And so this is, and all of these amazing photos here or screenshots are me, and I spoke with over 12 different leaders in philosophy with children, and this is us pondering. You know, when you speak with philosophers, you get to speak for a long time. So I had some amazing conversations with them. And in the end, I created a detailed research page of philosophy for children resources. I created 4 themed discussion curriculum plans. So these are things that people can use to help get conversations started with children. And I interviewed people on, like, what is philosophy with children? What are the current challenges? Where do they think the direction will go? And then focused on those research questions of what was the relevancy of doing philosophy for children, especially now. So that's a little bit of an overview of it. The first thing I want to say is, when I talk about doing philosophy with children, I don't mean teaching kids about Aristotle and Descartes and things like that. What it is it's a educational pedagogy, or so an educational tool that people can use to engage kids in critical thinking and just the ability to sit and question and wonder. So philosophy for children done well, what I learned is it's a really empowering tool for teachers to disrupt the traditional education dynamic. So in a traditional sense, knowledge is transmitted from a teacher to a pupil. And though this works for a lot of things, in many ways it can be disempowering. And so one of the things, you all can still hear me? Okay, perfect. One of the things that we learned about was that in order to create a good philosophy with children thing, you take about an hour, you bring together a group of K through 12 kids, you put them in a circle, and you start with the question, what do you wonder about? And the whole point is to allow kids to sit back and learn from one another. So the teacher doesn't have the answer, the teacher doesn't have a place where the conversation needs to go. What you do is you sit, the kids get to ask anything that they wonder about, and you slowly go through a relearning of how we can question, and teaching kids that just because you don't have the answer to something doesn't mean that it is a taboo question to ask. So one thing that I found in my research is that doing philosophy with children isn't necessarily more important during a time of crisis, but because of COVID-19, kids are home with their parents, and so the parents may be hearing the kinds of questions that kids are asking more often. Things like, why am I lonely? Why are people dying? You know, why are the police doing XYZ? All of these questions that us as adults maybe scare us a little bit because we don't know the answer to, and we've been taught that questions without answers are questions that probably shouldn't be asked. And so this is a tool that we can use in our education system to help kids learn that even the questions they don't have the answers to, there's still questions that have value that they can ask, and that the most important thing is to be able to wonder. The last little point I wanted to make was that I also learned during this research that we have a prejudice against technology especially. And what was really interesting when talking with people who were doing these philosophical conversations with kids over Zoom was they found that maybe it's because we're the internet generation, but kids were actually able to have really amazing and intimate conversations from their homes in a virtual setting, which was very surprising because a point of philosophy with children is that you are in a circle, you are with people. But they found that in some ways being in their own home and people having that kind of barrier of a screen allowed people to share things more intimately. And one of the examples is sometimes it's easier to say something by text than it is in person. So that was a really fascinating thing to see, is that philosophy for children can be brought into a more digital platform and be very different, but still be effective in giving children the space to be able to wonder, ask questions, and learn from one another. So yeah, I don't want to take up too much time, but did you all have any other questions about that or things like that?
Will you present these findings to, let's say, the 4J school system to start with? Very interesting.
Yeah. So the next plan is to sit in on more Zoom calls. Sit in on some in a elementary school in Hawaii. They have a really incredible philosophy for children program. And my next task that I will be doing is sharing these resources with local teachers and parents, and also with the UO education graduate school program. So that'll be the next step, is sharing that. But mostly it's a chance to, it's not a very well-known thing, doing philosophy with children, and in many ways it can be misunderstood. And so, yeah, the goal is to hopefully spread the word about the good that it can do, and give people, bring people to resources that they have in Hawaii and also at the University of Washington. But yeah, hopefully I will be able to do in person and try out these curriculums that I made in person, or at least on Zoom. Like, that is my main goal. So yeah, I'm so excited to be able to hang out with kids in person. That was I was so bummed that I didn't get to do, but soon.
Thank you. Well, all the more reason to continue this research, right? Thanks. Okay, so next, I think we have Haley Speed. Let's get you queued up to share your screen.
All right, one moment. I'm just scrolling through all of our folks.
Yeah, no problem.
Okay.
There's only one Haley, so it makes it easy.
You
Yeah.
should have the power,
Okay.
Haley. You go for it.
Let's see. Okay. Okay, let me present this. Okay, can everybody see the slide? Okay, so my project was called Functions of H3K27 Methylation in Neurospora crassa. So I know, really big, long title, but essentially we are studying how a specific type of gene silencing works inside of our model organism. And at the Selker lab, we use Neurospora crassa, which is a red bread mold. So kind of fun. It's like very pretty orange mold. But for those of you who maybe don't work in an epigenetics lab, I guess. Methylation is something, so like in this first picture, you can see this, there's this yellow strand, and that's DNA. And in order to fit inside the cell, because there's a lot of DNA and cells aren't very big, this DNA has to wrap around these proteins called histones. And sometimes the histones can have methyl groups attached to them. That's this red thing. And that changes the way that the DNA is expressed. So methylation will make it so that the DNA is less expressed, and therefore that it doesn't get used within the gene, because you don't always want to be expressing all of your genes at once. Oh my gosh. Okay. So essentially, I've been using something really cool to measure whether or not there is a methyl group on a specific gene by using tryptophan, which you guys might have heard of. It's like the chemical in turkey that like makes people sleepy. So tryptophan, one of the metabolic products of tryptophan. So when tryptophan is broken down, it makes anthranilic acid, which actually very conveniently is fluorescent. So you can see on these bottom samples here, these are tissue samples that have been given tryptophan, and some of them glow. This is our negative control. You can see this one does not glow. And the negative control is the gene that controls the breakdown of the tryptophan is methylated. So you can see this is a super easy way to tell what genes are methylated and what isn't. Just, does it glow, does it not glow? So essentially, in our lab, what my project is we have a mutant library. So we have a collection of, I don't know, maybe 13,000 different mutants that are put into 96 well plates. So we have 96 well plates. Oh, oops, okay. We have 136 96 well plates that each have different mutants in them. And I've been going through each of the 136 plates and growing up the strains and giving them tryptophan and basically seeing what happens. So this graph over here, you can see is an example of one of the 96 well plates that I did. So on the bottom here is how much they have grown. So it says absorbance. So how much, it's essentially like how much tissue we measured to be in the sample. And then on the top here is fluorescence. So most of the strains that you would see, like normal healthy strains, are probably going to be growing a lot and also fluorescing a lot. Because that means they're breaking down the tryptophan normally, everything is good. But what we are interested in is ones that grow but don't fluoresce, because that means that something is getting kind of funky with the methylation. So something like this, you can see it's not a super strong grower, but it is definitely growing and not fluorescing. So what I'm going through, I am going through the library. I actually just started plate 100 today out of the 136 plates. So I've got about 40 left that I still need to screen, but we found a couple ones that are interesting that I'll need to follow up on once I'm done with my initial screening. But if I do find something interesting, then I can figure out maybe why the gene that is knocked out in that mutant makes the methylation weird, you know, if it's something that we haven't found before. So I'm super excited to get through the rest of my screening and start looking at these guys, the ones that are really interesting. You know, maybe why aren't they fluorescing? Was it a fluke? Is it actually something, you know, that we're looking for? So with the future directions, it's just continuing screening and then following up on the strains that are interesting so far. So yeah, I'm super excited. I really like epigenetics, and that's where I'm on my project so far.
Very cool. And you have plenty of time. School doesn't start until Monday.
Yeah, I know.
Any questions for Haley, everyone? Myles? Was that a question or clapping?
I was just clapping for Haley.
Oh, okay.
Okay.
Okay, if we don't have any questions, I believe Anastasia. There was a question. I'm sorry. Go ahead.
Yeah, there's one in the
In the chat, do you know how tryptophan
Yeah.
works as a marker? Ooh,
as a marker.
question.
What kind of marker? Like how the tryptophan gets metabolized, or like how the anthranilate fluoresces?
Philip's nodding. Yes.
Yes?
Yes to all of
Okay.
the above.
Okay. Yeah, so like in a metabolic pathway, there's like several different things that something is broken down into. So if you start with tryptophan, there's maybe, 3 or 4 other like intermediate products, and then anthranilic acid, which is the one that glows, the one that I'm looking for. And so there are several genes that like catalyze the jump between those products, and a couple of those within the specific tryptophan pathway are marked by H3K27 methylation, which is the specific like brand of methylation that I'm studying. So if the methylation is messed up, then essentially the tryptophan just can't go through the pathway, and it's not going to become anthranilic acid. I hope that answered the question.
Thank you. Anyone else? Okay, thank you so much, Haley. Okay, who's up next? Anastasia Browning. Let's get you queued up.
Hi.
Hi.
Should be ready to go. Let me know if there's a problem.
All right, can everybody see it? Okay, so I'm Anastasia Browning, and I conducted the study with Professor Elliot Berkman. It's a big mouthful, the title of it. So basically we're looking at message framing amid the COVID-19 crisis. So the study was built on the theoretical models of approach versus avoidance framing, and also the concept of self-transcendence. In motivational research, goal pursuit is kind of considered to be a dichotomous system, and it's believed that how we frame our goals kind of has a direct effect on how well we're likely to accomplish them and like how timely we are. In one direction, you have approach frame goals, and these are kind of considered to be purpose-driven, active goals, and they're associated, sometimes have been shown to increase positive affect. So an example of an approach framing is, I'm going to study 2 hours a day to pass my exam. We're working towards passing the exam. It's a goal we want. And in the other direction, you have avoidance framed goals, and these are considered to be the avoidance of an unwanted or anti-goal. This is kind of an example of, I'm going to study 2 hours a day because I don't want to fail my class. You're working to avoid failing your class. Research involving message framing has also shown that avoidance framing is correlated with negative self-evaluations of optimism, depression, and other measures of well-being, as well as negative affect. When the coronavirus pandemic began to spread, the immediate messaging that we were seeing from the government, most specifically the CDC, was distributed using avoidance framing. The goal kind of was to protect yourself from contracting COVID-19. It's like use social distancing so that you yourself don't get COVID-19. I was immediately concerned that these messages would cause an already distressing and traumatic situation to kind of compound with further feelings of isolation, fear, hopelessness, stress. I was also really concerned because at the time, and kind of still today, this is all we have, this mitigating of the coronavirus to kind of help quell the spread of it. So it's really important that they were working and effective. So I also drew from recent research in self-transcendence theory, which suggests that the concern for the well-being of others, so kind of like an altruistic concern, increases an individual's responsiveness to new input. And that was kind of hopefully in an attack at kind of like designing a message that targets non-compliant individuals. And I started testing back in April. So this was kind of at the time the college age students, because at the time we didn't think we were going to be necessarily as affected by COVID-19 as others. And Yeah, I hypothesized that by using an altruistic goal in an approach framing of these protocols, instead of the self-protective goals that we were seeing in CDC messaging, we would hopefully be able to increase individuals' intent to follow the mitigation protocols, also decrease negative emotions and stress, and possibly increase self-transcendent attitudes. I'm sorry, this is all very wordy over here. But if you, going to our methods, we used Amazon's MTurk, which is a crowdsourcing site. And we had originally 862 United States participants, which we screened for folks who took less than 3 minutes. So it was in total 832 participants, so a pretty large sample. Mean age was 37, about 38. They were then randomized into 4 conditions, and each of them randomly saw a pamphlet, which we manipulated framing and goal orientation. So whether it was approach avoidant or self-protective or altruistic. They were then surveyed using self-report measures. We measured stress, affect, self-transcendent attitudes, intentions to follow mitigation protocols, pandemic attitudes, and then a battery of demographics and individual differences, just in case. I also added a behavioral measure at the end to test self-transcendent attitudes to see if we were kind of, able to bolster people's intention to care for others. And we did that by, we offered people the chance to either donate their earnings from the study to the Feed America's COVID-19 Defense Fund, or they were also offered the opportunity to keep it themselves. So for my analysis, I used a Kruskal-Wallis test rather than an ANOVA. And this was because we're not able to confirm the assumption of normality in our data distribution. For folks who don't really know what that means, it's kind of like a lot of it we work with in human research, we kind of expect this normal distribution, like say for stress, we expect that most of the people are in the middle, and then as it peters off to more stress, there's less and less, and as it peters off to extremely stressful, I think I got both sides there, but you're gonna see this kind of bell-shaped curve. But because of the nature of the pandemic, the data was highly skewed. People were very stressed. Mitigation procedures were also skewed, and affect actually tends to be skewed most of the time. So due to this, we used a Kruskal-Wallis test because it's non-parametric, and it allows us to work outside of the assumption of normality. And the results showed a significant difference of goal orientation for individuals' intentions to follow mitigation procedures. Specifically, self-protective goal orientation showed more reports of intentions to ignore mitigation procedures. So, for example, if they saw pamphlets telling them how to protect themselves from COVID alone, which they were, they were more likely to report an intent to ignore those suggested guidelines. So these results were highly concerning, obviously, seeing as the self-protective condition was the one that was most closely representative of the real world messaging that we're seeing. And this is again back in April. We also tested for age, gender, and employment status and religiosity, and all of those predicted intentions mitigation protocols, but there also remained a significant effect of this goal orientation when we controlled for them. In conclusion, our manipulations had no significant effect on perceived stress, affect, or self-transcendent attitudes, but these results definitely are a clear indication of the immediate need to kind of adapt our procedures and our protocol amid this global crisis. There's also some preliminary research out there that does show altruistic framing to significantly increase reported intentions to follow mitigation procedures, but the present study unfortunately did not confirm that. So obviously further research is needed to see what actually helps and is beneficial in our messaging to help kind of boost our intentions to follow these protocols. But in the meantime, it definitely suggests that we might want to reconsider what we are doing, if that makes sense. That's it.
Thank you so much. Does anyone have questions for Anastasia? No questions?
Well, I have a question.
Yes.
Yeah, so if you could, and you might have said it and I couldn't dissect the science speak, but if you could pick like, no, it was good, I followed mostly. But if you could pick like one I guess, method right now that you think would be most effective based on your research so far? Is there one that you would like kind of just randomly or like personally pick?
There's actually two that I think I would do to kind of continue on with the research. One is that I would probably adapt this to be a longitudinal study, not necessarily a very long one, but just one where we could kind of check at baseline to post exposure, like how well those intentions to follow mitigation practices actually track, possibly just like a 1 month journal of how often you've been washing your hands, for instance, you know. The other thing I would definitely do is our pamphlets. Actually, I'll share again, sorry, because I had one of them on here. So the pamphlet, if you see it down here, it was based off the CDC messaging pamphlets that they had on their website at the time, and they're pretty involved. It's like very, very descriptive kind of guidelines on how, like, what to do and how to follow the procedures. I think to make sure that we were specifically manipulating just an altruistic goal or just a self-protective goal, I'd probably have it down to like a 1 sentence thing, more just like follow these procedures to protect your community or protect your family, and maybe be a little more specific on that, just because there's a lot of room for wordiness to maybe kind of influence over necessarily what our goal was. So that might be why we saw differently than other studies on altruistic, not necessarily significantly promoting people's intentions.
Thank you.
Yeah.
Anyone else? Okay, so Julia Lowe should be queued up next to go.
Yes.
Thank you so much, Anastasia, and
Sorry.
all of you. I think that you have your posters ready for the Undergraduate Research Symposium in May. You just have to keep updating your data.
Okay, is this showing up on everyone's screen?
Yep.
Yes, okay.
Thank you.
So, hi everyone, my name is Julia Lowe, and this summer I worked on quantifying DNA damage in chromosome structure mutants. So, meiosis is a specialized cell division that results in haploid sperm and eggs, and meiosis is made up of 1 round of DNA replication followed by 2 rounds of cell division. C. elegans are a great model organism for studying meiosis and imaging due to their organization of the germline and transparent nature, which allows me to see all the stages of meiosis in order. C. elegans also have a marker for DNA damage in early and late meiosis. I chose to look specifically at early and late meiosis because this is where DNA damage is induced and repaired. So DNA is organized into chromatin on the chromosomes. Chromatin is tightly coiled DNA around histone proteins that all make up a nucleosome. There is heterochromatin and euchromatin. Heterochromatin is more tightly wound around the histone proteins, while euchromatin has more of an open nature. There are different marks that define heterochromatin versus euchromatin, and actually Haley in her presentation mentioned these as well. But for example, one of them would be H3K9 dimethylation, which is a heterochromatic mark. And this means that there were 2 methyl groups added to histone 3. The effects of chromatin states and loss of chromatin marks on DNA damage and repair and induction are not currently known, which led me to my research question. How does the loss of a specific heterochromatic mark, H3K9 dimethylation, affect the DNA repair system? So to answer this question, I looked at a MET2 null mutant, which lacks the H3K9 Me2 mark, but still produces haploid sperm and eggs, which allows me to assess the lack of this mark's effect on DNA damage and repair, and DNA damage, repair, and induction. So my lab mentor took pictures of germlines on a microscope, and then I was able to use Imaris to create surfaces out of the DNA in order to make them nuclei. And then from there, I created spots out of the DNA damage in order to quantify them. And this allowed me to determine how much DNA damage was associated with each nucleus during early and late meiosis. So once again, I looked at early meiosis because that is where DNA damage is induced. and late meiosis because that's where DNA damage is repaired. The blue in these images represents DNA and the yellow represents DNA damage. Qualitatively, my data shows that there's more DNA damage induced in the wild type than the MET2 mutant. In late meiosis, it seems to me that there is an equal amount of DNA damage in the MET2 and the wild type worm. The graph to the right represents my data from 10 gonads. So I had 5 wild type and 5 MET2. The white represents the MET2 mutant and the black represents the wild type. The green outline signifies early meiosis and the purple signifies late meiosis. The y-axis is the amount of DNA damage per nucleus. And the MET2 mutant starts out with less average DNA damage per nucleus, and this indicated to me that the presence of the H3K9ME2 mark is important in some way for the induction of DNA damage. However, in late meiosis, where the breaks are repaired, the MET2 mutant seems unaffected, indicating normal repair mechanisms in the absence of H3K9ME2. And you can see this on the right side of the graph where the averages are the same at the end of meiosis. So in conclusion, the MET2 mutant induces less DNA damage in early meiosis than the wild type worm. However, the MET2 and wild type have the same average DNA damage per nuclei in late meiosis. And this all leads me to the conclusion that the repair system is unaffected by the lack of H3K9ME2, but important for the induction of DNA damage. Some future directions for my research would be repeating the study with a higher sample size, creating surfaces out of the H3K9ME2 marks, looking at DNA damage in other chromatin mutants, or looking at other chromatin marks such as H3K9ME3. Thank you guys so much for listening today, and a special thank you to my mentor, Zach Bush, and Diana Labuda for being the primary mentor for everyone in my lab, as well as my fellow Labuda lab members for all of their support. And I would like to thank the FYRE coordinators and other funding received for this project. So thank you guys so much.
Thank you so much. Questions for Julia, friends? No questions? Okay. If you think of any, oh, was there a question? So if you think of any questions, just put it in the chat. So thank you so much, Julia. Our next speaker, who definitely does not have a virtual background behind her, is Noa Cohen. That's a beautiful background wherever you are, Noa. So are you queued up to share your screen?
Yes, I'm all ready.
Okay, excellent.
Sorry, it'll just be a second. I was queued up 2 seconds ago. As things go. Okay, here we go. Okay, can everyone see my presentation? Excellent. So as a few people had mentioned earlier, the COVID has exhibited particular challenges with human subjects. So my initial plan for my research project, which I would have conducted this summer, was to use around 30 participants who were chronic smokers who needed to see me for 10 sessions each, all within really close contact because they were using a neural net. So I'm still holding off that, potentially this winter we could do a modified version of this plan, potentially doing 5 sessions rather than 10. This project has been 1.5 years in the making, so I do want to talk a little bit about it. This summer has been continuing the training and continuing the preparation for Plan A. We have received IRB approval, in addition to crafting a Plan B research plan that is in the process of IRB approval, which is entirely virtual and will still have human subjects, but won't have nearly as much contact as Plan A. And then we do have a Plan C in the event that neither of those would work out, which would require no human subjects at all. So my Plan A, which I just received IRB approval for, which is really exciting, is reducing nicotine addiction through electrical brain stimulation. So attempts at smoking cessation for people who are chronic smokers frequently fail. As you can see from this graph, it is very, very challenging for people who are seriously intending to quit smoking, and a lot of rehabilitation techniques don't really work in the long term. So this is a global problem that is in desperate need of a solution. So Dr. Michael Posner, he's a professor of neuroscience here at the University of Oregon. A few years back collaborated with Dr. Yeon Tong, a professor of psychology at Texas Tech University, who is also an expert in meditation. He developed a meditation practice called IBMT, Integrative Body-Mind Meditation, which he proposed would improve areas in the brain associated with self-control and help people with cognitive functions, including mood, memory, and potentially even addiction. So together, they, in Texas, over 2 weeks, used human subjects, college students who were smokers, and had them for a half hour a day for 2 weeks perform IBMT along with Dr. Yeon Tong. And what was interesting about this study is Dr. Yeon Tong and Dr. Posner did not tell these college students that they are in a study about smoking cessation. Instead, they were told that this was a study about relaxation techniques and improvements in sleep. And so what they had found is that there was a significant reduction in smoking in the experimental group by around 60%. And I would just like to reiterate, none of these students had any intention to quit, nor knew that they were in a study about smoking reduction. Students that underwent a different meditation technique geared toward relaxation didn't show the same levels of reduction in smoking. And so this was a really exciting find, and MRI scans showed that there was an increased connectivity in the brain in the area associated with self-control. And what they saw were pretty significant white matter changes. And in these particular college students, there was actually a decreased amount of connectivity and white matter in the areas of the brain associated with self-control for those who had issues with self-control who were smokers. So upon further investigation, they found that when one meditates, there is an amplification of theta brainwaves in the brain, in the ACC, responsible for improved self-control. So brainwaves are electrical pulses from neurons communicating with each other, and theta waves often occur naturally when people are drowsy or in a sleepy state, and you can in fact induce it through meditation. And these theta brainwaves in the ACC were responsible for increased connectivity and ultimately an improved self-control in these participants. So onto my study. What I am interested in doing is seeing if I can induce theta brainwaves in the brain via a neural net. And the pink boxes here are which electrodes would be stimulated. And so in order to stimulate activity in the ACC, we would have people doing an attention network task. And we would have them do this for about a half hour over a day over 10 sessions. Ideally, those 10 sessions would be consecutive, but we would try our best to put our participants within a kind of a 2-week window. So additionally, we would also want to objectively measure smoking and subjectively measure cravings. So a way we would do this is via a CO monitor to objectively detect how much they've been smoking, and then additionally a craving task, which we'd also show them, I would say, about maybe 3 times over the total 10 sessions, the first session, the middle session, the last session, to see whether they are continuing to crave cigarettes and smoking. So that is my plan A. Fingers crossed that I can start doing experimentation in the winter. But as you can see, it is very involved. There are a lot of human subjects that are not going to be U of O students involved because all of our participants will need to be chronic smokers. So we'll see. I'm continuing my training and making sure that I'm very familiar with the equipment and, you know, neural net acquisition, and hopefully in the future we'll be able to do it. There is an exciting new equipment that we might be able to use, which is a headband that our participants might be able to put on themselves. It wouldn't be as sensitive as a neural net, but that's a potential way to make sure that I'm keeping a safe distance from the participants. So anyway, on to plan B, which is something that we've developed over the summer, is rather than using electrical stimulation to increase the amplitude of theta, instead we will be using auditory theta to see whether it improves brain connectivity. And so auditory theta is otherwise known as pink noise, which would be a sound that we would play over the computer. And that would increase the amplitude of theta brainwaves in the brain. And we're interested to see if it has a similar effect as electrical stimulation. There is a lot of promising data with mice, and we're hoping that's going to transfer over to humans. And what we're going to specifically test are improvements in mood, which is going to be via like a questionnaire about people's general feelings that day. We'll also test improvements in memory, which will be done through a visual working memory test on the computer. And then also an intention network task, which also was going to be used for Plan A, which is essentially kind of like a computer game that activates the ACC and will help us measure people's improvement in attention. And so we would ideally have you know, anywhere between 50 to 200 participants, depending on the time that we're allowed to conduct these experiments. And we would invite them for 2 sessions. The first session will be, you know, we'll split our group into 2. And so there will be an experimental group and there will be a control group. In the first session, one group will undergo white noise stimulation, which is not supposed to be effective. And then another group will undergo pink noise, which is the auditory theta. And then in the second session, it will be reversed to see whether there is actual improvements in real time in mood, memory, and attention. So hopefully Plan A or Plan B can take effect sometime this year. If neither plan works out in the end, I do have a Plan C, which involves a literature review looking at lesions in the brain and seeing whether potential electrical stimulation can assist people with strokes. So yeah, I'll keep you all updated.
Thank you, and congratulations on getting the IRB. That's a big step, right? That's a big step. Anybody have questions for Noa? Okay, next up is Lucy Roberts.
I said awesome, but I was on mute, so you guys didn't get to hear it, but I said it. Okay. Here we go. So over the last summer, I have been working with Dr. Sutherland's Ocean and Ice Lab to do quantifying of iceberg movement and melt in Greenland's Thermalik Fjord. And so what the Ocean and Ice Lab has been doing for the past 8 years is every year they've flown out to the specific fjord in Greenland and put GPS tracking units on icebergs that are floating through it. And so what ends up happening is a fjord is an inlet from the sea, and in places like Greenland that have glaciers, the glaciers feed into the fjord. And right here is the point where they break off. And then as they're flowing out, this is the really dense icy area known as the melange. And then they end up hitting more of an open water, which is the fjord right here. And Dr. Sutherland's team has been putting GPS trackers on icebergs while they're in the melange, and then following those icebergs as long as the GPS units last, which can be anywhere between like 2 weeks, and I think we had one last over a year. And so from that, we can tell how the ocean currents are moving in these areas and how the icebergs are distributing fresh water within these saltwater areas. And so for the first half of the summer, especially, I was working on kind of updating our data. It was each year takes on like a different data form. So like condensing it all into the same data file within MATLAB. And then they had previously published a analysis of this iceberg flow using just data from 2012 to 2013. And I was looking at how that data changed when you added all 8 years into it. And as we moved into the second half of the summer and we were looking for ways to kind of wrap the research up and then continue to move forward, we focused on the second half of that research proposal, which was adding in remotely sensed imagery to our toolbox on how we can analyze this iceberg movement data. And so what we did, or a lot of different organizations, including the US government, the European Union, and then other private organizations have satellites that are orbiting the Earth. And every 2 weeks take really high resolution images of everywhere on Earth. And so this is an image produced from the EU. And all of these images are available for the public to download. It takes some technical knowledge of how to use it and to analyze it. And so what we did was take this, and then you have to adjust for the reflectance. So how bright the sun was at that day and like what angle it's at will determine how these icebergs show up on the images that are produced. So once you adjust for things like reflectance, you get these really clear images. And if I wasn't on Google Slides, I would be able to zoom in and you can very clearly see the boundaries of where all of these icebergs are. So these red markers down here are the actual markers of the icebergs that had trackers on them in this year, which was 2019. And so because our GPS units were reporting about every hour, it's very accurate to where the iceberg was at the time this picture was taken. I think within 15 minutes, we have a reading for the GPS unit. And so from that, I can draw outlines on the icebergs and be able to determine how big they are and estimate how deep they are, so we know how much they've been melting since we have images for every 2 weeks. And so right now, as I'm wrapping it up, what I've been working on is creating a spreadsheet basically of all 8 years and all of the images that we have for all of them, and then which icebergs are visible in those images and how big the icebergs were and how much they've been melting between the images that we have.
Very cool. It looks like a gigantic cell to me with dendrites coming out of it.
It was pretty fun. I was supposed to go to a conference on human rights in England that got canceled like 2 days before I was supposed to go. And so I keep like joking with myself that at least like I get to be spending my summer staring out the plane windows and
Thank you so much. Anybody have questions for Lucy? Thank you. Okay, next up is Deanna.
Hello. Okay, let me get set up. All right. Can everyone hear me? Yep. Okay. We can all see. Okay. Great.
Yep. You're good to go.
Fantastic. So I'm Deanna Plunkett, and I'm from the Huxtable Laboratory. And my research this summer focused on how neonatal inflammation disrupts microglia in brain regions controlling breathing. So, of course, the brain controls breathing. And within the cortex, we have functions like sensory processing and motor control. And then in the medulla, in the brainstem, we have the nuclei that are responsible for generating respiratory rhythm. This communicates to the motor neurons in the ventral cervical spinal cord, which innervates the diaphragm, allowing you to breathe in and out. A threat to respiratory control is inflammation in the brain. Brain inflammation undermines breathing, and the cells that are responsible for resolving this inflammation, allowing you to breathe normally again, are cells called microglia. Microglia can resolve inflammation in a multitude of ways. They can send out signals to other cells, they can change their shape in order to consume other cells, and they can divide, thus increasing in number. And when they're doing these things, they're considered activated. Microglia go through this activated stage early in development before becoming ramified or going into a resting state, so that in adulthood, when there's a normal adult immune challenge, they can become activated, resolve the inflammation, and then return to resting. But neonatal inflammation can prime microglia. So neonatal inflammation occurs in this early developmental period when microglia are activated, and it forces them to stay activated into adulthood, so that when there's subsequent adult inflammation, they become overactivated. And this is concerning because overactivation of microglia changes behavior. So we're wondering, does priming impair brain regions that control breathing, which is vital to life? So my hypothesis is that neonatal inflammation increases microglia sensitivity to, specifically microglia in brain regions that control breathing, to subsequent inflammatory stimuli. So we test this by first inducing neonatal inflammation, aging these animals, and inducing adult subthreshold inflammation. We induce subthreshold inflammation because healthy microglia will not react to subthreshold inflammation. And we then analyze if there's an increased microglia sensitivity by determining if microglia themselves have increased. And we can determine this by immunofluorescently labeling microglia and counting them with flow cytometry. We first have to establish an adult subthreshold dose of inflammation, and we did this by testing 10 doses of lipopolysaccharide, or LPS, which is what we use to induce inflammation. We analyzed the percent of microglia after these doses in the medulla and spinal cord, these regions that control breathing, and we determined something that we call the half maximal response. This is when there's a 50% increase in microglia percent. We found that this occurred at 25 micrograms per kilogram in the medulla and 59 in the spinal cord. Thus, we chose 24 micrograms per kilogram as our subthreshold dose of inflammation. So we found that neonatal inflammation plus adult inflammation in the medulla increases microglia percent compared to our controls, which had neonatal saline plus adult saline or plus adult LPS, and compared to neonatal inflammation plus adult inflammation. We did not see the same pattern in the spinal cord, but we did see this again in the cortex. This increase in microglia percent in animals that experience neonatal LPS plus adult LPS indicates that neonatal inflammation may increase medullary microglia sensitivity to subsequent inflammation. So this means that early life inflammation may prime adult microglia, and these changes in microglia may impair neurons that control breathing and thus increase sensitivity to inflammation. So these adults who experience early life inflammation may be more susceptible to impaired breathing during an otherwise insignificant inflammatory stimulus. And this is only preliminary data with an N of 1, so moving forward, I would like to continue to increase my N and thus further support my results. So thank you so much. I'd like to give special thanks, of course, to the entire Huxtable Laboratory, and specifically my PI, Dr. Huxtable, my mentor, Sarah Baylor, everyone at the CURE team, and all my fellow SURF and FYRE peers, and funding for this was provided by an NIH R01 grant.
My name's Alexa, and this summer as a FYRE student, I ended up going on a road trip through Oregon and Nevada looking for hot springs. So there's a huge database that the Bureau of Land Management has made where they have listed all of the GPS coordinates of all of the hot springs that either that have existed at some point. So what I ended up doing this summer was I took the big long list of all of the hot springs that were recorded in Nevada and Oregon, And I was looking at, basically my role was, can we find all of these hot springs and are they usable to collect biological samples from? So the overarching question that my lab wanted to ask, and I was a part of the Luca lab, was, can we find some information in taking biological samples from these hot springs to shed light on some of the more poorly understood aspects between how these microbial communities may have evolved and how that is directly related to the local geologic conditions. So like nearby fault lines. So as I mentioned, I ended up going on this huge adventure this summer. We had 106 sites that we needed to locate. Down in the bottom left-hand corner, you can see it was me and my partner, Sage Albright. He is also a part of the Luca lab, though he just recently graduated. It was a week long, and what a typical day looked like for us was up at 4 a.m. We were trying to beat the July desert heat, so that's why we got up so early. And then once we were up, we had to pack our campsite. We were tent camping along the way, eat a quick breakfast, and then we were hitting the road. I had to plan our entire route from beginning to end. So I put them all in an order that seemed to make logical sense for efficiency. And then what we ended up doing was seeing how many we could get done in a day. That way we could be fluid. So some of these hot springs were completely dried up. Some of them we couldn't find. So we had to be incredibly flexible with what we ended up finding that our circumstances were on the road. And when we got to each hot spring, what our job then to do was to take photographs of the hot springs, as well as measurements on like how big the spring was or what the temperature or pH of these hot springs were, so that we can narrow down these 106 hot springs to a very small handful to then return to again in the future and collect biological samples. So the process of collecting samples requires tons of equipment. You have to pack a big dry shipper and hike with it out to your site so that when you collect your samples, you can store them in a liquid nitrogen chamber so that they stay frozen and preserved until you get back. But that method is only sustainable for a certain period of time. So the purpose of this pilot survey was to be able to narrow down the ones that are inaccessible or not worth going to. That way, when we return, we can be really efficient and collect those samples and get back out. So in the pictures, I have some photos of a few of the, in my opinion, cooler hot springs that we found. What you can see around the edges of them are these sort of orange colored, when you're there in person, it looks a little bit like rubbery, kind of foamy. And there's a close-up in the middle underneath that hot spring picture of what the mats look like. So we were looking for a really strong presence of this orange bacteria. And that's how we were kind of figuring out, is this somewhere that we can come back to? Also, while we were out, we had to be very, very careful about safety conditions because these aren't your typical Yellowstone hot springs. They don't have ropes and chains telling you where you're not supposed to walk. And so Sage and I had to come up with a really good system of communication where we would let each other know everything we were seeing and hearing, from like the way the ground sounded when you walked on it to, do you see holes up ahead that we should steer around? And then also there were just things like rattlesnakes and whatnot that we had to look out for. So it was very, very useful in the sense that now we were able to report all of this back to our research professor and say, okay, here is realistically what these sites look like, and here's information that you can't find online about them. So it's giving us a really good start. This will end up being a multi-year study across the Midwest, so it won't just include Oregon and Nevada. We're hoping to expand into Wyoming and Washington and California. So our timeline's obviously gotten pushed back because of COVID, but we were able to get quite a bit done this summer, as 2 people way out in the Oregon and Nevada wilderness is pretty socially distanced, I would say. But yeah, so if anybody has any questions, I'd be happy to answer them.
I have a question. How did you gauge the heat before you, because I can imagine it must be a little bit like a minefield, right? Without signs and things like that, like when you go to Yellowstone.
Right. So there are 2 pictures, or 3 pictures, I guess, that kind of show the equipment that I was using. So what I'm holding in that bottom left-hand picture is a painter's pole. And on the end, we fashioned a cup onto the pole that would be able to swing. And the idea was that we could extend the painter's pole, dip into a hot spring from far away so we don't have to get too close, and then we pull it back, and then we'd put our temperature, pH, and conductivity probe inside of that cup. What we ended up actually doing is we found it was more practical to use the painter's pole and the probe as more of a pulley system, because the cord is super, super long and meant to be, I guess, used from a distance, that you can drop it down into very, very deep holes. So what we did, and you can see this in the bottom sort of towards the middle, I'm crouching over a big crack in the ground. This is a hot spring that was just very, very deep and we couldn't very well access, as we used the painter's pole to support the cord for the probe, and then we hung the probe over the side and slowly lowered it like a pulley down into the hot springs. And then we just went just below the surface to protect the probe and not expose it to too much, because these hot springs were very, very hot. These box and whisker plots are a little bit difficult to see because they are so small, but our average temperature we were finding was about 74 degrees Celsius, which is about 165 degrees Fahrenheit. So just trying to minimally damage our equipment. It is meant for that, but as well as try to provide as much safety to ourselves as we could.
Thank you. Any other questions for Lexa? Thank you so much. Yeah, Anastasia.
You might have said this, but just I'm rereading over your conclusions, and you picked the best 18 sites. Is that just kind of like the most variety of bacteria, the most substantial amount, or is that what considered those hot springs the best ones?
So these 18 are probably actually going to be narrowed down even further. 10 is more realistic for being able to collect enough biological material from each and ship it back. But these are the 18 that were safe to approach and actually work with close enough that you could scoop material out of them. They were big enough that we knew, since we were there in the middle of July and they still had decent size, we knew that they wouldn't dry up at other parts of the year. And these are all ones that had, the biggest concerns were just safety and accessibility. They were the most safe, most accessible. We knew they'd be there year round, and they had strong microbial growth. So we were just looking for those mats. And if we could find all of those things, it made the list. And then going forward, we'll probably narrow it down based on which are the most similar in temperature and pH, so that they're the most comparable as we move forward.
Very cool. Thank you.
Cool. Thank you. Any other questions, friends? Thank you so much to everyone for taking the time to join us in the middle of the day. Yes, great job from Professor Labuda. Yes, I agree. Great job. And please know that your connection with us does not stop here. We are and always will be here for you for resource guidance as you move forward in your academic and professional and postgraduate trajectory. Thank you.
Yeah, it's just been a privilege working with all of you. Again, our third cohort of summer undergraduate research fellows and our inaugural cohort of first-year research experience fellows. And so, as Lance said, we're here to continue with you on your journey, all things undergraduate research, graduate school, distinguished scholarship. So we are, again, it was wonderful to get to know you this summer, and we look forward to continuing to work with you. So thank you so much. And all being back together on May 27th, 2020. Yes. At the Undergraduate Research Symposium.
Yes, exactly. Yes, if we don't call on you for other events before that, yes, we will definitely all meet up again May 27th, 2021. So put that on your calendars now. Thank you, everybody. Have a great week, and best of luck with the start of school for everybody. Thanks again.
Thank you.
Thank you.
Bye.
Bye.
Bye.